some discussion, let's say questions. So if students don't ask, then I have some questions for the students and we can like discuss the answers and have some sample questions.
Perfect. All right, everybody's getting in, settling in. Welcome, everybody. Another live class here at Atlantic International University. So we have a new lecturer with us today who's bringing in new topics that are going to range, let's say between chemistry and biology, and then branch off from there. So there's a lot of things. I think I'm going to allow Paula Abola to introduce herself and to give you a sense of who she is. So Paula, take it away.
Yes, so hi everybody. My name is Paula. I come from Latvia, but I live in Germany. And I have a background starting from chemical biology and medicinal chemistry. That's where I got my bachelor's in. And then I moved on to studying my master's in clinical drug development, which led me to pursue my PhD in clinical research, focusing specifically on Parkinson's disease. Because Parkinson's disease is actually genetic in my family. My grandmother had it. So this is something that I want to pursue and actually potentially maybe find a cure. And yeah, so I've started teaching recently at university in Germany as well. And I'm really happy to have this opportunity here at Atlantic International University and hopefully work together with you guys and not only teach you, but also learn something from you as well. Okay.
That's perfect. So they're muted. They can't actually activate their microphones.
Okay.
Unless they raise their hand, then we can ask them to activate their microphone, and then they can ask you a question. Do you want to take questions during the presentation, or would you prefer just waiting till the end for all the questions?
It depends. If somebody wants to clarify something, they can ask throughout as well.
Perfect.
Yeah.
All right. I think we're ready to get started.
Okay, perfect. So good morning or afternoon, depending on where you guys are, and welcome to our first lecture on chemical biology. And I'm actually very happy to have you here today. And I will start off by saying that if you've never heard of chemical biology before, you don't have to worry because you're in the right place, because we're going to begin with laying a strong foundation for the rest of the course if we continue this. And we'll do this by answering three essential questions today together. Firstly, what is chemical biology? Secondly, why it's important. And thirdly, how it is used in research and medicine and actually solving real world problems. And you might notice that chemical biology is around us every day. So this is something we're going to look into as well when we have our discussion questions. And maybe you might not notice that every day, but we interact with it every single day. And as said, this lecture is designed for those who may have also no background in chemical biology, and I'll try to explain everything in clear terms and also give examples and analogies as we go through these ideas and make sure that we understand them together. So let's move on. So what is chemical biology? To understand chemical biology, we need to begin by breaking down the term in its two parts. So we have chemistry and we have biology. Chemistry is the study of matter, meaning the physical substances that make up everything in the universe. And in chemistry, we focus on how atoms and molecules are arranged, how they interact, what reactions they undergo, and what properties they have. And chemistry helps us understand things like why salt dissolves in water, how batteries store energy, or how certain materials conduct electricity. Whereas biology focuses on living systems, on living organisms, and it's concerned with how organisms live, how they grow, adapt, and how they function. And biology looks at how cells operate, how tissues form, how organs interact, and also how everything works together to sustain this life. So no matter if it's a bacteria, an animal, a human, so how all these systems operate together. And you might ask and wonder what happens when we combine chemistry and biology. And this is what comes to chemical biology. It's the discipline that uses chemistry as a tool to investigate biological questions. So it's actually really interesting. And it's more than just studying what is already there. So chemical biology also aims to influence and manipulate biological systems through chemistry. And it's a field that sits right at the interface of the physical and life sciences, especially with drug development these days, because a lot of chemical biology is used to understand how our biological systems work and actually make targeted therapies and targeted drugs for certain diseases. For example, also cancer actually is one of the leading fields right now. And I think that one important question that raises from this is then how is chemical biology different from biochemistry? And maybe you guys know that biochemistry is more about analyzing the chemical processes that happen naturally inside living things. For example, we have a biochemist who might isolate an enzyme from a cell and study how this enzyme works in a test tube. But the chemical biologist would go one step further and use chemical techniques to actually intervene in this process, and often inside the living cell, to actually learn something new or to achieve a certain result. So they're not just going to analyze what already happens to a certain enzyme or a protein, but they're going to try to use it and do something further with it. And a simple example to show the contrast would be, suppose that we want to understand how a particular protein functions inside a cell, and a biochemist might extract the protein, purify it, and run tests on it inside the laboratory. But the chemical biology might design a fluorescent molecule, a fluorescent dye, that specifically binds to that protein inside the living cell. And when it binds, it will glow up and it will actually allow us to watch how the protein behaves and where it is located in real time under a microscope. So this is the added layer that chemical biology brings to science, and it brings the chemistry into the biology in a very active and actually hands-on way. So, okay, and why is chemical biology important? So now that we have the working definition of chemical biology, the next question actually is, why does this field matter? And chemical biology plays a critical role in helping us understand life at the molecular level and in solving major problems in healthcare, biotechnology, and beyond that as well. So first, it allows us to explore how life works at the smallest possible scale, at the level of atoms and molecules. And every biological process, whether it's your body digesting food, your brain forming a memory, or your immune system fighting off a virus, it's fundamentally driven by interactions between molecules. So chemical biology gives us tools to study those interactions in very great detail, which helps us better understand how our bodies function and how things go wrong in disease, for example. Secondly, chemical biology is key to developing new medicines. As I already mentioned, it's being used in drug development, and many modern drugs, including treatments for cancer, infections, and genetic diseases, were made possible by research in chemical biology. And by designing and testing molecules that interact with specific proteins or genes, we can develop therapies that are more targeted, more effective, and safer. You might have also heard of the CRISPR gene editing being used for cancer treatment and other genetic disorders. So this is also a very good example of chemical biology and how it's brought these more targeted therapies, more personalized therapies for people who are struggling with these genetic and very complicated diseases. And thirdly, chemical biology has revolutionized the way we visualize and diagnose diseases. So advances in chemical biology have led to breakthroughs in imaging techniques, such as fluorescent microscopy and PET scans. And these technologies let us look inside these living cells or even inside the human body to see how disease develops and how the treatments are working. And we can also track disease over time and track how the treatment, if it's functioning properly, if that's the way we want it to be. And finally, chemical biology enables us to build entirely new biological systems. And this is where the field overlaps with synthetic biology. For instance, scientists can engineer microbes to produce useful substances such as insulin, biofuels, or biodegradable plastics by rewriting their genetic instructions. And these custom designed systems can then perform tasks that nature never intended, giving us powerful tools for industry, medicine, and environmental protection. And this is why it's also important that we bring this chemistry into the biology, because as you know, as we've just defined, biology is all about the living organisms, whereas chemistry can bring in the chemical part for the biology. And this is why we can do things that nature never intended. And this is something that is very important. And to give you a concrete illustration, for example, when penicillin was discovered in the 20th century, it happened by accident. And it was actually just a lucky find. But today, thanks to chemical biology, we don't need to rely on chance. We can design antibiotics in the lab, and we can do that with precision and with targeted individual goals, let's say, put it that way. And we can create molecules that selectively attack bacteria while sparing human cells. And we can do it based on our understanding of molecular structure and function. And this is the power of this field, because we don't need to be first giving these antibiotics to humans and then seeing what happens. And I said we can make them these molecules that will selectively attack bacteria. And why it's also important in this infectious disease research is because we know that we have a lot of antibiotic resistance, which is a very big problem. We have a lot of these antibiotics that are not working anymore just because they've been misused or overused. And this is why it's very important that we can create molecules that will actually selectively attack bacteria and potentially have a new field of antibiotics. And then we're going to look at core concepts of chemical biology. So if we look at these central ideas that define the field of chemical biology, then these concepts are the building blocks that we're going to come back to again and again throughout the whole course. So the first key concept is that chemical reactions are at the heart of biology. Everything that your body does, from moving, thinking, healing, growing, it involves a series of chemical reactions. And these reactions are obviously not random. They're carefully controlled by enzymes, which are biological catalysts. And enzymes make these reactions happen faster and more efficiently than they would on their own. For example, if we consider a group of enzymes that are called kinases, these enzymes have the job of attaching a small chemical group called a phosphate group to other proteins. And this process, called phosphorylation, because of the phosphate group, can turn proteins on or off, which in turn controls many essential functions inside the cell. For example, growth, communication, and repair. So it's like flipping a switch. We're saying it's going to be on or off. And the second concept is molecular recognition. This refers to how biological molecules identify and interact with one another. And most molecules in your body have specific shapes, just like puzzle pieces, like if you think of a jigsaw puzzle. And for one molecule to affect another, so whether it's a hormone binding to a receptor or an enzyme locking into a substrate, they need to fit together in a precise way. And this is also often referred to as lock and key. As you know, every lock has a specific key. So in our bodies as well, we have an enzyme, and for it to lock into a substrate, they're going to look like lock and key. So certain enzyme cannot lock into every single substrate. They need to fit together in order to do that. And a good example here is hemoglobin, which is the protein in your red blood cells. Hemoglobin binds to oxygen molecules in the lungs and then carries them through your bloodstream to tissues all over your body. And the ability of hemoglobin to recognize and bind oxygen depends entirely on its molecular structure. And this is why only hemoglobin can carry oxygen around your body, and another protein cannot do that. So another protein will in turn have its own function. So this is why they are unique in a way. Okay, and the third concept is the use of chemical tools. Chemical biologists develop and use molecules, which they call as probes, to explore these biological systems, as I already mentioned in the definition of chemical biology. And these probes can help identify where a protein is located, whether a certain gene is active or how a disease is actually progressing inside a human body. And one famous tool, which you might have heard of already, is the green fluorescent protein, or known as GFP. And when scientists attach this GFP to another protein inside a living cell, it makes that protein glow under UV light. And this allows researchers to track the movement and behavior of proteins in real time without disrupting the cell's normal functions. And it's obviously a very incredibly powerful window into the dynamic world inside living organisms. So we're able to, in real time, track what happens inside a cell. So that's something that had never been done before chemical biology. And this green fluorescent protein technique can also be applied in click chemistry, which is a part of chemical biology, because you can imagine clicking this fluorescent protein to another protein and allowing it to go inside a living cell and then glow under UV light and actually track it in real time. And if we think about the real world applications of chemical biology, then firstly, let's take a closer look at drug discovery and development. So in the area of drug discovery, chemical biology helps us find new compounds that can be used to treat a disease. And by understanding the structure of disease-causing molecules, then we can design drugs that specifically target them. So it's almost like designing a key to fit a specific lock. Again, coming back to the lock and key mechanism. And I think this is very important in cancer research and also, of course, in other diseases, especially in genetic diseases, because at some point we had no understanding of how exactly this disease is progressing inside a living organism. And now when we're able to do that, we see what proteins or enzymes are involved, then we can develop these drugs that will specifically target them. And we have also an example of an anti-cancer drug, Gleevec. And this drug was actually designed to block a protein that drives a certain type of leukemia. And because Gleevec was developed using chemical biology tools, it's extremely specific and it attacks the cancerous protein, but leaves all the healthy cells largely unharmed. And this specifically leads to fewer side effects and better outcomes for patients. So this is something that we do not see with, for example, traditional chemotherapy. Chemotherapy attacks also our healthy cells. And this is why in cancer research, chemical biology can be very revolutionizing and actually lead to better patient outcomes and actually, let's say, improve the quality of life for patients with cancer, because a lot of time these traditional chemotherapy treatments was something that, you know, was bad for the patients, and their quality of life would suffer not just because of the disease, but also because of the treatment they were getting. And now chemical biology allows us to make sure that it attacks only the cancerous protein, but leaves all the healthy cells mainly unharmed. Of course, we're not saying that there aren't some side effects, but this is why also people might be getting less side effects, such as losing their hair. This might not be happening with these new anti-cancer drugs. And the second real-world application is medical imaging and diagnostics. So chemical biology has given us tools to visualize the invisible. And one such tool is positron emission tomography, which is PET scanning. You probably heard of that as well. And this method uses specially designed radioactive molecules that are injected into the body. These molecules accumulate in areas of disease, for example, in cancer cells, and then emit signals that can be captured and translated into detailed images. And this obviously allows doctors to locate tumors, monitor treatment progress, and detect relapses early. So you've often probably heard that patients with cancer undergo PET scanning at certain times during or after their treatment. So this is all for the doctors to see if we see the tumor growing or shrinking, hopefully shrinking from a treatment. And this way we can monitor the progress and we can also detect if the relapse occurs. So this is a very also important thing. And as you can see, this chemical biology comes a lot back to cancer, because cancer is obviously cell biology. It happens to our cells, and this is why it's very important. And I think that soon it can also revolutionize actually cancer treatment and cancer diagnosis. And further, we have synthetic biology and protein engineering, where chemical biology helps us design new biological tools from scratch. And one of the most widely used applications is the production of insulin. In the past, we know that insulin was extracted from the pancreases of pigs or cows, and the process was very expensive, inefficient, and obviously risky sometimes. But now scientists can insert the human insulin gene into bacteria, turning them into like miniature factories that produce insulin safely and cheaply in the laboratory. And this is possible because chemical biology gives us control over genetic and biochemical processes. So this is something that we are able to now do in the laboratory instead of using pigs or cows, which obviously is very expensive and can also bring certain risks, certain diseases. We even know that there were some animal diseases from pigs or cows transferred to humans through this process. And this is why chemical biology has now allowed us to just produce insulin by inserting an insulin gene into bacteria. Then we're going to talk about experimental techniques in chemical biology to explore biological systems effectively. Chemical biologists rely on a variety of advanced experimental techniques. So I'm going to briefly introduce three of them that we're also going to encounter later in the course. So we can talk about it in more detail in further classes. But the first one is fluorescence microscopy, which is a quite a popular one. And this technique allows scientists to look inside living cells using fluorescent tags. So as I said, these fluorescent dyes. And these glowing tags will help us observe where the proteins are going, how they're interacting, and what changes happen over time inside a cell. So for instance, scientists will use this fluorescence microscopy to track how cancer cells invade surrounding tissues, providing insights into how tumors spread, so how metastasis happens. The second technique is mass spectrometry. You might have heard of that as well. And this method allows us to identify and measure the exact composition of molecules inside a sample. And it's especially useful for studying proteins and small molecules in biological samples. So if we can use mass spectrometry to detect early warning signs of diseases like Alzheimer's even before the symptoms begin. Because obviously, as also the name implies, mass spectrometry, it's going to identify and measure exact composition of certain molecules in a sample. So it will give you the mass from which you can actually say what certain molecule it is. And this is obviously important because we could use it on a sample. For example, if we suspect that a person might have increased risk for Alzheimer's, we might take a sample and we're going to measure it, and we find a certain molecule that is related to Alzheimer's disease, then we know that these are the early warning signs, even much before the symptoms are going to begin for the person. And thirdly, we have the CRISPR and genome editing. So I mentioned CRISPR briefly, but it's actually a revolutionary tool that allows scientists to cut and edit DNA at precise locations. And this opens up the possibilities for treating genetic diseases, also improving things like crops, or creating model organisms to study disease. And one promising example is using CRISPR to correct the mutation that causes sickle cell anemia. So they've started doing that. And as we know, sickle cell anemia is a very dangerous genetic disease. And they've started using CRISPR to cut DNA at certain points and replace it with a healthy genetic sequence that actually corrects this mutation, which has then caused the sickle cell anemia. Also, this could be potentially able to cure a lot of genetic cancers. I think they've started talking about it in breast cancer. We know that breast cancer is often caused by a certain genetic sequence. So if we can edit that sequence and replace it with a healthy gene sequence instead of the mutation, then we could potentially also cure breast cancer in that way. And this, of course, also applies to other genetic diseases like genetic forms of Parkinson's disease, what I've been studying as well. And then we, of course, also have challenges in chemical biology. So while it has already achieved a great deal, there are, of course, still major challenges ahead. And one challenge is selectivity. So when we design a drug, we want it to only affect the intended target, but not other molecules. Like I also said about cancer treatments, that it would be good that it attacks only the intended target, for example, a protein, but it's going to leave all the other cells healthy. But we know the biological systems are complex and molecules can sometimes bind to the wrong place, which often leads to side effects. And this is something that needs to be considered regarding chemical biology and going forward. So selectivity and side effects are a very important thing, especially in drug development. And another challenge is, of course, the complexity of biology itself and biological systems. So living systems involve thousands of interacting components, and making changes to one molecule can actually have ripple effects throughout the whole system. And this can lead to unintended consequences. So this is something that brings a challenge into chemical biology, because our systems, they interact in every single way, from cells to tissues to organs. Everything is an interaction, and this just changing even one single little molecule can affect the whole chain down the line. And the third area of growth, and of course also challenge, is the integration of AI. And AI is now being used to predict how molecules behave, to design new drugs, and of course analyze big data sets. And this combination of AI and chemical biology is still in its early days, but it holds enormous promise for speeding up discovery and making research more efficient. So we might be able to bring a drug out of the laboratory into human clinical trials much quicker than we were able to before. So looking forward, we can expect that chemical biology will play a major role also in personalized medicine, where treatment will be tailored to the specific individual or patient, and also in creating new synthetic systems for addressing health, environmental, and industrial problems. And then I want to take a quick look at the history of chemical biology, which is quite interesting. So chemical biology goes back to 1828, where scientist named Friedrich Wöhler, a German scientist, made a groundbreaking discovery. And he managed to synthesize urea, which is a molecule found in our urine, using a non-living chemical reaction. And this shocked the scientific world because it challenged the belief known as vitalism, which held that biological substances could only be made by living organisms. So Wöhler's experiment showed that life's building blocks could be created through chemistry alone. And this is actually where they started researching chemical biology and how chemistry can actually not only help biology, but even be a part of it and be integrated in it. So because he managed to synthesize a purely molecule that is found in living organisms, we were able to understand that it can be done just purely from chemistry. So there has to be some sort of way to bring the two together. And so why was it an example of chemical biology? As I already mentioned a little bit, is because it bridged the gap between chemical and biological worlds. So Wöhler used a purely chemical approach to study a biological molecule, which was urea. And in this way, he bridged the gap between chemistry and biology, showing that biological molecules could be created through chemical synthesis. And it laid the foundation for later discoveries in biochemistry and molecular biology as well. And other key figures in chemical biology include Louis Pasteur, who discovered molecular chirality and demonstrated that fermentation was driven by enzymes and not just spontaneous processes. So he discovered that biological molecules could exist in different mirror image forms, which is known as chirality. leading to the development of stereochemistry in biology. So we often have molecules that are actually mirror images of each other, and this is known as stereochemistry. And he also showed that fermentation was driven by enzymes. So he linked chemical reactions to biological processes. And further, we also have Emil Fischer, who studied how enzymes interact with their substrates. And he introduced the famous lock and key model that I mentioned earlier. So he studied carbohydrates and proteins using chemical synthesis. And he developed the lock and key model of enzyme function, explaining molecular recognition. So that a certain enzyme is going to lock in with a certain substrate, and this enzyme will not be able to bind to another substrate that belongs again to another enzyme. And then we also have Linus Pauling, who applied quantum chemistry to biology and helped us understand the structure of proteins. And he applied quantum chemistry to study biological molecules and particularly the protein structure. So he helped establish chemical biology as a modern scientific discipline as well. And in summary, chemical biology is exciting and, of course, interdisciplinary field that uses chemistry to understand and manipulate living systems. It is also a very challenging field, and it continues to grow. So it's really hard to keep up to date with all of it, of course. But it plays a vital role in medicine, of course, in diagnostics and synthetic biology and drug development. And we looked at the key concepts, powerful techniques, and as well as real-world applications that make this field so dynamic. And we've also seen how its roots stretch back nearly 2 centuries, which reshaped the way how we think about life and biological systems that we can still apply chemistry to as well. And for the theory part, that is it from my side today. But I have prepared also some sample questions for you guys, which we could discuss. So maybe Dr. Lambert, how would you like me to take this further?
How do you have the questions? Do you want to just say them, or do you have them in your presentation?
So I have them in my presentation.
I would say have them in your presentation, show them, and then the students can answer in chat.
Okay.
Are they multiple choice?
No, they're like more discussion questions. So I don't know, can they unmute themselves?
They can raise their hands, like we already see CC Jules has already raised his hand. Do you see that?
Okay, yeah, I see it on my other screen. Yeah, I do.
Yeah. So
Okay.
what we do is there's a little, if you hover over his picture, you'll see a little ask to unmute.
Okay, let me see. Ask to my,
And you
okay,
can
yes.
click on that and say, okay, CC Jules, go ahead with your
Okay.
question. There you go.
Yes, CC Jules, go ahead.
Okay, very quickly. Thank you. A very good presentation. So very technical and very professional.
Thank
So
you.
now my concern is about 3 questions, maybe together to be summarized in the answer.
It looks like
The
the
first one
questions are
is
already written into chat.
yeah. Okay. So you can read in the chat to go fast, no problem. Instead, I talk. It can take time, so much time. Any interaction between chemical biology, 3 core functions, as well as chemical reaction, molecular recognition, and chemical probes and tools. If there is any single interaction between the 3 core of chemical biology.
Well,
second question. Okay. You can do question by question, answer question by question, or
yeah, let's take one question at a time.
Okay.
Yes. So if I understand your first question correctly, you mean that these 3 core functions that we discussed, you want to understand if there's one thing linking them all. Is that correct?
Yeah.
So as said, not in a way, not really, but at the end of the day, we come back to biomolecules. So we come back looking at biomolecules, and we know that these molecules undergo chemical reactions. And of course, we have this molecular recognition where we're using chemical biology to see how things work. And for that, we're also using the chemical probes and tools. So it all comes back to molecules. So I could say that that is the one that brings chemical biology, these 3 core concepts together, if that makes sense.
Okay. Question 2. Are there common function, chemical biology based between the three field application as well as the drug discovery and development, imaging and diagnostic, synthetic biology and protein engineering? And what are pros and cons of these chemical biology field applications?
So you are asking about the common functions between drug discovery and development, imaging and diagnostics, and synthetic biology and protein engineering. So as you see, this is, again, comes back to medicine, to treatment, to patients of certain diseases. Well, as I also mentioned, we're using chemical biology to do targeted drug discovery, targeted drug development. And for that, for that as well, if we're using this new drug now in a certain patient, then it's important that we can use this imaging to track how this drug is behaving and if it's actually treating the patient, so we can see the disease progress. And of
what
course,
are the pros and cons?
Yes, and of course, pros and cons. Well, I think I mentioned some of the pros of chemical biology in itself, that we can have these personalized medicines, so targeted treatments. Whereas cons, again, we have a lot of confounding variables, even demographics of patients, where then we have to make sure that we're applying it to all sorts of populations, meaning from different ethnicities, from different age groups, and that can, of course, be a con as well.
Okay. Now, the last question is, since I'm in the measure AIML, artificial intelligence machine learning, so I have a concern, very, very relevant concern about the application of chemical biology in AI application, which is, have you any idea on the algorithms, which is based on the AI application for chemical biology? Because
So,
we have
yeah.
clustering, we have key means, we have many algorithms based on which AI application running for any field.
Yes, so that's a really good question. I will admit that I am not such an expert in AI and I'm not aware of the algorithm, but as mentioned, this is a growing field and this is something that I actually want to look into myself as well. But I will admit that I cannot help you here with the algorithms, but it is an interesting field for me too. So that's something I'll be looking into next.
I will look for. Anyway, I will look
Yeah.
for.
Well, maybe not so much algorithms, but when you try to work with AI, you have to give it information, ask it questions, and see if it can come back with some answers for you.
Okay.
And I think the idea is to help you. It'll give you some ideas to work and innovate. You can innovate some new ideas with it.